Good afternoon, everyone. Welcome to today's UCLA Clinical and Translational Science Institute Distinguished Speaker Seminar. It is my pleasure to introduce our speaker, Dr. Lihong Wang, who is the Bren Professor of Medical Engineering and Electrical Engineering at Caltech and the member of the National Academy of Engineering. His laboratory's focus has been centered on photoacoustics, a physical phenomenon that converts absorbed optical energy into acoustic energy, allowing for a hybrid imaging modality that acoustically detects optical absorption contrast. This provides a powerful technology for non-invasive multi-scale biochemical, functional, and molecular imaging from organelles to humans at high resolution. Dr. Wang's group has pioneered innumerable aspects of this technology, including in vivo photoacoustic tomography, photoacoustic endoscopy, and photoacoustic reporter gene imaging. In addition, his group developed compressed ultrafast photography, leading to the world's fastest real-time camera at 70 trillion frames per second. Dr. Wang has published over 500 peer-reviewed articles in journals including Nature, Science, Physical Review Letters, and PNAS, given over 500 keynotes, plenary, or invited lectures. His book entitled Biomedical Optics, Principles, and Imaging was recognized by the Joseph Goodman Book Writing Award. Dr. Wang chairs the annual conference on photons plus ultrasound and was the editor-in-chief of the Journal of Biomedical Optics. His laboratory has been funded by the NSF and NIH and received multiple awards, including the IEEE Biomedical Engineering Award and the Optical Society Michael Feld Biophotonics Award. A quick housekeeping item: use the Q&A tab to type questions that we will address at the end of the lecture. Please join me in welcoming Dr. Lihong Wang, whose lecture is entitled World's Deepest Penetration and Fastest Optical Cameras, Photoacoustic Tomography, and Compressed Ultrafast Photography. Dr. Wang.
Thank you very much. May I share the screen? I'm not allowed to share right now. Okay, let me try it again. Slideshow. Great. Thank you very much, Dr. Packer, for the very kind introduction. Let me thank Dr. Jiang Shai for inviting me to give this talk as well. I'll be talking about two aspects of the research in our lab. One focuses on penetration into scattering biological tissue. The other one is focusing on speed. And the first technology is called photoacoustic tomography, and the second one is called compressed ultrafast photography. White light, we have a lot of imaging modalities already. Optical imaging is very special because it gives us molecular specificity. From physics, we know that light-matter interaction is uniquely positioned at the molecular level. Given the fundamental roles of molecules, we really have to work on this technology, despite all the challenges. We can provide in vivo functional imaging, very much analogous to functional MRI, in vivo metabolic imaging, analogous to PET, in vivo molecular imaging of gene expressions or disease markers. We can even provide in vivo label-free histologic imaging of cancer without tissue exclusion. However, we face daunting challenges because turbidity is ubiquitous. And you see all materials are scattering by nature. If you look at biological tissues in our body, with the exception of the eye, most biological tissues are. We need to overcome the turbidity to gaze into biological tissue. Right. There's something wrong with my timing. Let me restart. So, hold on a second. All right, let me share the screen. If you look at photon transfer, light transfer in biological tissue, you will see something like this. Initially, the photons travel as a group but within about a millimeter or so, the photons are scattered all over the place. The laser propagation is very much like a diffusion process of dye or heat that you might be familiar with. So that presents a huge challenge in optical imaging. Now, if you look at the history of optical imaging, it starts from about 300 some years ago. We started with conventional microscopy that allows us to provide very high resolution, but the penetration is limited to 100 microns, which is the so-called aberration limit. We stagnated at this limit for hundreds of years until modern light sources were invented, which allowed us to get confocal or two-photon microscopy or optical coherence tomography, enabling us to advance the penetration by order of magnitude up to about 1 millimeter, which is the so-called optical diffusion limit. By using photoacoustic imaging, we advanced the penetration by nearly 2 more orders of magnitude. Now we're talking about multiple centimeters of penetration and still provide you very high spatial resolution. We do face the next challenge, which is called optical dissipation limit. Which technology can overcome the next challenge is still unknown question, unknown. Now, photoacoustics is a known physical phenomenon since Alexander Graham Bell reported this phenomenon in 1880. However, he had no access to laser or other modern concepts. So in his days, he used the sunbeam to implement this concept. He encoded sound into a light beam, then propagate the light beam. The remote end can receive the light, convert the light back into sound again. So you can hear actually directly. Now again, because he had no access to the modern concepts of laser, ultrasonics, tomography, and computer, we actually combine this set of modern concepts with pretty old physics to form a new imaging modality called photoacoustic tomography. In photoacoustics, we broaden a short laser pulse, make sure we're within the safety limit. So the NC safety limit on laser energy per area we can deliver to humans without causing any damage. We allow photons to scatter around to provide deep penetration. When light is absorbed, it generates very rapid heating. Every millidegree gives us a detectable signal already. So this is acoustic emission due to the thermal expansion. Then we deploy ultrasound transducers outside the tissue to receive the ultrasound signal induced by the light absorption. Because acoustic scattering is orders of magnitude weaker than optical scattering, we can form a very sharp image. However, the contrast is from light absorption. So we can see molecular information at acoustic resolution for very deep penetration. And this is the fundamental reason for the combination of light and sound in a single modality. We do have to use reconstruction method to form an image. Without getting into the detailed math, let me use a simple analogy to explain the reconstruction behind this method. So let's say we want to image a single sound source, like a thunderbolt. When you see the lightning, you have time 0. And when you hear the thunder, you can record a time delay T1, multiply T1 by the speed of sound. You can get a radius for a spherical shell on which lightning took place. Now, if you have 3 such measurements centered at different locations, we can triangulate and pinpoint the exact location of this thunderbolt. Now, in biological tissue, of course, we have a volumetric sound source. It's much more complex than a single point source. So we oftentimes use hundreds, even thousands of ultrasound transducers to form an image. The detailed math is called the inverse spherical read-on transform, very much analogous to the linear read-on transform used in X-ray CT, except we have a higher dimension, we have a curved integration surface. In 2003, we demonstrated the first functional, also the first in vivo photoacoustic tomographic images in small animals with an intact scalp and skull. And so by wiggling one side of the whiskers, the contralateral site was activated. We can see the activation using our technology. This paper has become one of the most cited papers in our field. In fact, since the publication of this paper, you can see the exponential growth of our field. Since 2010, the conference on photoacoustics has become the largest at the 20,000 attendee photonics. And now it's the largest by far. Our field, since a few years ago, has started publishing over 1,000 peer-reviewed papers per year. And you can see this, there's a pandemic dip. Hopefully we'll pull out of this very soon. This technology has been commercialized by a number of companies. We should highlight that Seno Medical has received FDA approval for human breast imaging last year. I'm associated with several companies myself as well. So fast forward to 2017, we developed the first single impulse panoramic photoacoustic computed tomography technique. For the earlier work, we used a single element transducer. We had to rotate around the small animal to form image, which took about 20 minutes to form image. That's obviously too slow to be practical. And with a single impulse, we're much faster. We can configure the system for truncal imaging, start with a laser, route it through a conical lens to form a hollow cone beam. We use a 4-ring ultrasonic transducer to get a full view. That's so-called panoramic view. And we're only showing half of the ring here. We follow the engineering principle of nyclid sampling to minimize motion artifact, to minimize any subsampling artifacts. We use 512 ultrasound detection channels. And we use a preamplifier to boost signal to noise ratio, to conserve the signal to noise ratio in order to avoid cabling noise. And we use a 1-to-1 data acquisition for the highest speed data acquisition. Within a single laser shot, we form a cross-sectional image. The data acquisition is finished within the acoustic transit time across the field of view, which translates for a small animal into tens of microseconds. That's how fast we can form image. So you can appreciate it. So there's no motion artifact to worry about here at all. Tens of microseconds, the motion is so small. Then, of course, we use a computer to reconstruct image and present the data. Here's a close-up. You can see the hollow cone beam. Here's the ultrasonic focus for elevational resolution, and we run the image reconstruction for natural resolution, XY resolution. The system can be reconfigured for brain imaging. Here we use a diffuser to form a solid cone beam, and then the rest of the system is very similar to trunk. Just to give you one example, here we can scan in real time at 50 Hertz. Each frame is acquired within tens of microseconds. Then we repeat our laser pulse to scan along the trunk at 50 Hertz. So we have 50 frames per second with the current setup. And you can see the cross-sectional images here. Right now it's showing the kidney. You can see the symmetrical structures of blood vessels. Here we acquire images without injecting any contrast agents. So everything is done. And you can even see the heartbeat right here. And so our technology has been licensed to a company. Of course, our first demonstration was a few years ago, and the company has actually improved the technology so much, their image quality is actually better than our previous image quality acquired using our system. And this is one example where they are looking at dynamic deposition of IgA protein in renal cortex. And I understand this is a cardiovascular group. We can actually image the rat heart using this technology. This is one example where you can see the heartbeat. Using our technology, you can get quite detailed map of the heart. Of course, as soon as we demonstrated small animal imaging, we decided to scale up the system for human imaging. This is a human breast imaging system. We call this single breath hole panoramic photoacoustic CT of human breasts. And you can see the system is bigger now. This diameter has to accommodate most breasts. We decided to start with 22 centimeters in diameter, which can accommodate most breasts, more than 95% of breasts. By slight deformation, you can compress the breast to less than 4 centimeters. So this is not a painful compression, very different from X-ray mammography. And so we're compressing against the chest wall, where X-ray mammography is a sideways compression. That's quite painful. So ours is much more natural. And the rest of the system design is very similar to the small animal system, except that we scale up the whole thing. This system operates at this point. It gives us a few centimeters of penetration, hundreds of microns resolution. Now, one advantage of photoacoustic tomography is the scalability between imaging depth and spatial resolution. And each bar represents one implementation of this technology. And of course, there are many, many versions. I'm only showing you a few representative versions here. And this dashed line connects all the different modalities, different implementations. And so this shows the scalability between the depth and resolution. The ratio is roughly constant. And that means that it doesn't matter which depth you target, you can expect about 200 pixels in the depth direction. And so that presents high quality images for us to work with. And so this type of scalability should play an important role in future biology, because we want to connect information acquired from all relevant length scales, from organelles all the way to small animal organisms or human organs, and in a coherent way, because the contrast stays constant. We were looking at light absorption, molecular information as our contrast. No matter what length scale you work at, you have the same type of information. So it's very natural that we can combine across all length scales. In fact, photoacoustics can also serve as a central glue to combine with all other imaging modalities, because photoacoustics is naturally compatible with ultrasound imaging. We use ultrasound transducers for detection, where pulsar ultrasound use them for both transmission and receiving. And we share some contrast mechanisms with MRI, as I will demonstrate later. And in fact, we can even use some sort of labeling that gives you PET contrast. So you can see how well we can integrate with other modalities. This is one of the first images we acquired from a healthy human volunteer. You can see the breast. We use color to encode depth information. So starting from the nipple region, we get a single shot 2D image within 150 microseconds. Again, you don't have any motion artifact. Now for 3D imaging, we want to minimize motion artifact. We ask the patient to hold their breath. Within 15 seconds, we can acquire a 3D image. We can also compress it to 10 seconds for special patients if they wish. The smallest blood vessel diameters we've imaged is about 1/4 of a millimeter in size. And so you can get much finer small vessel images than even MRI. Now with MRI, oftentimes we require injection of gadolinium, which is a heavy metal contrast agent. So we're doing contrast agent-free imaging. The maximum penetration is about 4 centimeters, as demonstrated in this slice by slice images. Now, of course, the next goal is to image breast tumor, breast cancer. And so this is a demonstration of a case that's radiographically dense. So even for this 69-year-old woman, and the breast was radiographically dense, X-ray mammography missed the tumor, but ultrasound picked up the tumor. We labeled the tumor location with a metal target on the skin surface and took a second mammogram. And using our technique, we can see the tumor region quite clearly, as shown by the double arrows. And we can analyze the density of blood vessels, which stands out. And we also get elastography for free. So we can look at a stream map, because we can image so fast, we can track the motion of blood vessels. And due to the natural motion, we found out this tumor region is stiffer than the surrounding normal tissue. Again, because we can image it fast, with fine enough resolution, we can identify blood vessels and detect which is artery, which is vein. And so what we do is we look at the dynamics of 2 of the vessels, for example, and you can see here the magnitude of their motions are very different. And so obviously the one with greater motion represents artery. We can color code them. Then we decided to advance our technology even further. So this is a human breast / brain photoacoustic tomographic system. And so you can see the system was originally designed because it was funded for breast imaging, but we want to double the system for brain imaging. And you can see this schematic right here. We use 2 different modes to operate. One we call structural or anatomic mode, which takes about 10 seconds to form a 3D image. But the functional mode is faster, it takes only 2 seconds. Now we have 1,000 channels, 1024 channels, as opposed to 512 in the previous system. And there are two different laser wavelengths. 1064 offers oxy-dominant contrast, and 694 nanometer offers deoxy-dominant contrast. So, and this is again quite different from bold MRI, which is mainly sensitive to deoxy only. And we have 4 different arcs that allows us to scan the system very quickly, rotate it very quickly to cover the entire hemisphere. And this is an example breast image. You can see we can get nearly isotropic images of blood vessels within the breast. And this is a projection type of image. You can see how well we can see blood vessels using this technique. This is the first demonstration of the first imaging of human brain. So with finger tapping, you can see brain activation in this region right here. And wait for the next cycle. Again, you can see brain activation with the finger tapping right here and here. That's the first step right here. So we started with this special patient group, which is hemianatomy patient. So the skull was temporarily removed from the fingers. On the next slide, I will let our collaborator narrate what we observed.
Here we present results from subject 1, 7T MRI on the left, functional pact on the right. Individual functions are flashed in, starting with finger tapping, lip puckering, tongue tapping, passive listening, and silent word generation. Vessels for co-registration are then labeled 1 through 4, vessel 1 and 2 being superficial temporal artery, 3 and 4 being cortical arteries. The images are overlaid, and then individual functions are flashed in, starting with finger tapping, lip puckering, tongue tapping, passive listening, and silent word generation. The vessels are again pointed out individually, vessel 1 and 2 being branches of the superficial temporal artery, 3 and 4 being cortical vessels. The lateral perturbations allow for visual spatial appreciation of the co-registration. The images are then separated, and native T1 masks are faded in. Again, 7T MRI on the left, functional pact on the right. Individual functions are then flashed in, starting with finger tapping, axial, and coronal images through the area of interest are introduced below. Next, lip puckering, tongue tapping, passive listening, and silent word generation.
As you know, 7T MRI represents the strongest B field strength FDA approved for clinical use. So we're comparing with the state-of-the-art MR machines. The big question is, why do we need photoacoustics for brain imaging? Now, in comparison to functional MRI, photoacoustics provides richer contrast because we can detect both oxy and deoxy very quickly and provide functional imaging. Our response time is shorter in comparison to bold, and the background is much lower. The linearity is higher, much greater portability. It's an open platform, lower acoustic noise, much lower cost, because the bold MRI system at 7T costs about $10 million, and it's also very expensive to maintain. We don't use a magnet. We don't have to worry about bringing surgical tools that are magnet compatible. Now, of course, that very first demonstration was done without skull. The next goal is to demonstrate with implant. And we call this trans-implant functional photoacoustic tomography of human brains. And so despite the implant, which is acrylic, we can still see functions. Here, this is the initial demonstration of finger tapping response. Then ultimately, we want to see through skull. So this is the first demonstration. For the same craniotomy patient, we look at the left side, which is hemicraniotomy without skull. Then at the same time, we look at the right side through skull, because the right side is normal. And you can see the finger tapping responses on both sides. So both are detectable. This is extremely encouraging, of course. Where's the ultimate limit? And this is what's exciting about photoacoustics as well. Physiologically, the functional response is actually far shorter than the current reported time we can detect. It's a few seconds right now. And it turned out we can push to sub-second, as demonstrated in a small animal. And this is a single impulse stimulated fast initial dip in a single capillary level of mouse brains. And so we look at fractional change of our signals with a single impulse of only 40 milliseconds long. And we can see onset of oxygen saturation within 130 milliseconds. That's how fast ultimately we can potentially push once we refine our technology. Of course, we face daunting challenges because here we're looking at a microscopic level in a small animal in an invasive way. But non-invasively, the signal to noise ratio would be much harder to get. Now, if you were to look at the total hemoglobin response, it's a slower response. It's about 3 times as slow, as you can see right here. As I said earlier, that photoacoustic imaging can be scaled to microscopic level. This photo shows the first 3D photoacoustic microscope, which was built by our lab. Without getting into the details, let's just look at the key components here. And so we have this optical illumination that's focused into the tissue. On the tissue surface, we have a donut beam. The core of the beam was made dark to minimize surface interference. The ultrasonic detection is confocal with light illumination to maximize the signal to noise ratio, so you can form a good image. With a single laser shot, the time of arrival of the signal will give us a 1D image in the depth direction. Now, if we raster scan the head in the water tray, we get a 3D image. We have a membrane window here that couples light and sound. The tissue is placed below the membrane window. This system now operates around this point. I jumped from here to here. So this is microscopic level imaging, but the penetration is scaled down as well. So in this particular version, provides about 3 millimeter penetration, the tens of microns resolution. We're looking at a representative image of our own palm without injecting any contrast agent. If you want to image blood vessels using X-ray, most likely we'll inject iodine-based contrast. And here we just use blood as our natural contrast agent. We can see blood vessels quite well. And you can look at the B scan into the skin. So that gives you different layers of the skin structure. We also want to use this technology for imaging of circulating tumor cells. As we know, there's no standard technique that allows us to see CTCs in vivo in humans. And we first pushed this technique to the clinic to image melanoma CTCs, because melanoma CTCs tend to have a higher concentration of melanin loaded in the cells. And so for the first time, we can actually demonstrate that those CTCs may actually show up as a bright spot in this particular patient. You can see this bright spot appears several times. And here in this example, you can see a doublet that's captured multiple times within our field of view as well. And so this may have lots of applications in terms of maybe screening, diagnosis, monitoring, therapy, you name it, or even prognosis. Because we can image so fast, potentially we can examine the entire 5 liters of blood in humans. And so that allows us a better accuracy even than drawing blood, because drawing blood may have sampling error. Now we can push the technology even further for higher resolution imaging. So now we're talking about maybe about 1 millimeter penetration, that's single-digit micron resolution. That allows us to get cell level resolution. And this is done in vivo. You can see single cells and watch how they bifurcate. And we can image humans. This is the image of our own finger cuticle. And we can monitor the oxygen saturation of hemoglobin. And we see the color to represent oxygen saturation as cells release oxygen. And you can see the color change. So hopefully we're providing a tool for physiologists to study oxygen delivery at the most fundamental level. We all know that pathology is still the gold standard for disease diagnosis, but it sometimes can be really slow. The question is, can we make it faster? Can we make it label-free? So here, we're providing label-free photoacoustic histology by imaging the DNAs and RNAs in cell nuclei directly. And this is a photoacoustic image without staining. And we can see the entire tissue area and differentiate normal from tumor regions. And we're looking at a breast tumor, human breast tumor right here. In fact, our pathologists can make diagnosis out of our images. And of course, to validate, we compare with the standard HC stained images. You can see how well they correlate. And so we are looking at DNAs and RNAs directly without staining. Arguably, this might be even more accurate because HC staining has labeling errors as well, because some of the DNAs and RNAs may not be labeled properly. And this is another example of a bone specimen. So bone pathology is extremely challenging because you have to decalcify. That whole process can take up to 7 days to finish. But using our technology, because we don't have to decalcify, we can just image the bone directly. And we don't even have to smooth the surface because we can accommodate the surface profile. And we can finish the entire scan within 11 minutes. And this is not even the ultimate limit. And you can see how well the two images compare. And here we use artificial neural network to pseudo color our images. So pathologists don't even have to be retrained to read our slides. Now we can push the resolution even further to the organelle level. And this is one example where we image mitochondria. And now, anytime you use a wave to form an imaging method, you're limited by something called a diffraction. And so the diffraction limited resolution is 234 nanometers. However, using nonlinear photoacoustics, we can go beyond this diffraction limit and provide photoacoustic nanoscopy. This is also called SuperResolution Imaging at 90 nanometer resolution. You can compare the two versions and see the drastic difference between the two resolutions. And here's an EM micrograph for comparison. Now, let me move on to the other modality we've been working on. That's called compressed ultrafast photography, or CUT in short. And so, as we know, we live in 4-dimensional space, X, Y, and Z, and time T. So this is working on the time aspect. Now, standard imaging modalities are not fast enough to image light pulse, for example. As we know, light pulse, light propagation is the ultimate speed, if Einstein is still correct, because nothing can propagate faster than the speed of light. And so using our technology, operating at 100 billion frames per second, we can see a light pulse for the first time in real time. And so we don't have to repeat the laser pulse, just fire the laser pulse, we can capture the entire event in real time. That's playback with 10 billion times slowdown so we can watch. We're literally watching the slowest slow motion movies now. And you can see the light pulse propagating in space gets reflected, refracted into a different medium. And there are 2 light pulses here having a race. And we can look at fluorescence. And in fact, we developed our system into a 2-color version. The green channel captures the excitation light, you just saw it. And the red channel captures the fluorescent light. And so this is a single shot fluorescence imaging. And some of you might have heard of FLIM, fluorescence lifetime imaging microscopy. And this is essentially a single shot fluorescence lifetime imaging without repeating the event. So you can watch potentially biological events in real time. Now with FLIM, standard FLIM imaging, you have to repeat the event. And so that takes quite some time to build up the image. That's much less than real time. Maybe I can quickly go through how the system works. We start from the object with intensity I versus xy time and time t. And so we route the intensity to this digital micromirror device, which provides a code C, it's a pseudo-random number code, and allows us to encode I. And the encoded intensity or light is routed to this street camera. And so unlike standard street cameras, we open up the slit as widely as possible, where standard camera uses a very thin slit. So that basically removes the white dimensional information. So here we maintain the vertical dimensional information as well as the X dimensional information. The photons are converted into electrons through the photoelectric effect. We apply a voltage ramp to shear the electrons. Depending on the time of arrival of the electrons, they are sheared into different deflection angles. And so that's a very important encoding process, because now we freeze time, so to speak, because the deflection angles can be later on translated into time. And this represents very high temporal resolution. Then we convert electrons back into photons. We capture the photons again using a CCD. The entire physical process can be represented using a matrix. And the CCD captures this energy matrix versus XY, and the intensity goes through the coding process C, shearing S, and time integration by the CCD camera T. All we have to do is to solve this equation. However, we have a problem because the recorded matrix is two-dimensional, the intensity I is three-dimensional. So you have more unknowns than we have measurements. So, the high school teachers will tell us this cannot be solved. But now we have a compressed sensing concept. And so the true number of unknowns in I is actually less than the number of measurements. So using this compressed sensing concept, we can solve this inverse problem and form a movie. There are a number of applications. One of the first one is to detect superluminal Mach cone, and this has never been observed before. In sound, you have this supersonic Mach cone or sonic boom. NASA captured this image. So when the object flies greater than the speed of sound, and you will see this sonic boom. And when light pulse propagates faster than the speed of light in the medium, you can see optical boom as well. So this is a single shot capturing of this event, and this is related to Cherenkov radiation, which has medical applications. Phase contrast is important for biology because some cells without labeling, you don't really see the cell structures very well. And if there are fast events that provides phase contrast, but not amplitude or intensity contrast, we can potentially use our technology to see it. And this is a demonstration where we see a shock frown using the phase contrast. Another demonstration is Kiosk. Kiosk has important applications in biology as well. The heartbeat irregularity, for example, fetal hypoxia, they tend to be associated with chaotic modeling as well. And so maybe one day we can find some application in biomedicine. And for now, we demonstrated using physics. So we have this cavity, half mushroom-shaped cavity is a chaotic cavity. And so you launch a pulse into it. We took two different pulses. So those are two different takes. And if you merge the two, initially, you'll see that they travel together. But with time, they will split. And so that's a signature of the Kiosk. Here, we even study the transition of the conversion between a stable cavity and a chaotic cavity using a light speed switch. And so we start by launching the photon packet into the stable cavity, then we open up the gate and it'll transition into the chaotic cavity. So the technology is fast enough to monitor any chaotic phenomena that may not be so repeatable, so the standard technique may not work. If you compare our technology with all the standard high-speed modalities, and so we're plotting the sequence depth, meaning the number of frames you have per movie versus the imaging speed. The preferred corner is here, and the cup family sits around here. Now, just because it's fast, it doesn't mean we cannot slow down the cup camera. We can just slow down and fill up the entire horizontal axis. Let me thank my lab and Caltech first of all. I'm very grateful to Caltech for building me a dream lab. And this photograph shows one of the floors of the lab. And we have omniscale imaging, microscopic, nanoscopic at this end, and the distal end presents human imaging. We have openings for graduate students and postdocs. Credit goes to the lab members. We're mainly funded by NIH, but we have some funding from the NSF. We have a couple books if you're interested in some details. Thank you very much. I'll be happy to entertain any questions.
Thank you, Dr. Wang. Thank you so much for your presentation and in-depth review of these two fascinating stories. It was very, very enlightening. A few questions on the photoacoustics. So you showed very elegantly how the application for breast imaging is now already FDA approved. And then you showed very convincing data where you're comparing 7 Tesla MRI versus photoacoustic imaging in different stimuli activating different areas of the brain. So my question is, does it not matter then in terms of penetration, whether you're going through bone, muscle, air, or adipose tissue for this technology?
Actually, light will be attenuated when we go through tissue. The deeper we want to penetrate, the weaker the light will be. So that does represent a challenge. And so in human breast, so far we demonstrated about 4 centimeters from each direction. So if you were to deliver light, you can potentially double that thickness to 7 to 8 centimeters. In fact, some labs have demonstrated penetration or in terms of tissue thickness, you can image on that level. So the skull represents another level of challenge as well, because skull is not ultrasound friendly. The good news is that skull, it looks white. That means it doesn't really absorb so much. It just scatters light. Scattering doesn't really kill photons. So they just reroute photons around. Photons will just bounce around. So we can send light through the skull and reach the brain cortex. But the sound coming out will be aberrated. The wavefront will be distorted by the ultrasound. And so that's actually a deterministic phenomenon. So there's hope that we can de-aberrate and form sharp images. And this is an ongoing problem. Now, even without de-aberration, we can still form image, but it tends to be blurrier than the images acquired without skull. And so that's a great question. This is a very active and exciting area that we have challenges to overcome as well.
Thank you, Dr. Wang. So we have 2 questions by Sami Yanikian pertaining to photoacoustics. The first one is, how efficient is functional stroke mapping with this technology?
We have not tried that yet. Stroke mapping. So right now for brain imaging, we are providing a couple of centimeters penetration into the brain, and so that's deep enough to image the brain cortex. But stroke, I guess, can take place anywhere in the brain. So sometimes it may be beyond the penetration limit of our current technology. However, our lab is also interested in microwave-induced acoustic imaging. And so microwave can provide much deeper penetration, or RF. As we know, MRI is actually based on RF. And so you can provide essentially whole body type of penetration if you use the right frequency. And so for stroke, maybe that's a better technology.
Thank you. And the follow-up question is, in terms of therapeutics now, I guess, could this technology, meaning photoacoustics, be used for stem cell stimulation? And the example provided is stem cell stimulation of hair cells in the cochlea to improve hearing perception.
And that's a very interesting question. So of course, for photoacoustic imaging, we want to make sure we operate in the safe region. We follow the NC safety limit. The pressure we induce is very, very low, much lower than the ultrasound imaging. Where in ultrasound imaging, you send a pretty strong ultrasound pulse. Of course, that's also within the safety limit. You send strong ultrasound into the tissue, only a tiny fraction is bounced back. That's what we detect in post-cycle ultrasound imaging. Maybe roughly, on the order of 0.1% of the energy comes back. In photoacoustics, the acoustic signal we produce is actually very similar to that level. And so very similar to the detected ultrasound level. So we're extremely safe. So the question is, do we need light to generate sound? If you can use piezoelectric transducers, you can generate sound directly. If you want to stimulate stem cell, that's one option. Unless you have some optical selectivity you're targeting, through protein level, you want to selectively excite a given area, that's definitely a possibility. And there are ongoing studies, people have been trying to use photoacoustics or maybe photothermal effects to stimulate neurons. That is available.
Fantastic. Thank you so much. Thank you, Dr. Wang. So I think we have one more question. By Dr. Mack. So the question is, utilizing blood vessel imaging and extraction of stiffness, can you quantify changes in vessel compliance? For example, atherosclerotic plaque progression.
That's another great question. We haven't demonstrated that, but based on the signals we've demonstrated, I think that's a possibility. We can see the magnitude of the change that's directly related to the compliance of the blood vessels. That would be fantastic. For that version, right? We can image quite deep.
I think there's a lot of excitement. Yeah, there's a lot of excitement based on the questions on the potential therapeutic applications also of this technology.
Right. So of course we demonstrate in the breast, but there are other places we can study and maybe push into that direction. Yeah.
Sounds good. So I think we have no more questions in the Q&A box. So Dr. Wang, thank you so much for your presentation. Thank you for taking the time to give the CTSI lecture. We do hope to have you back on campus in person in the near future.
Thank you so much for the invitation.
Thank you, Dr. Wang.
All right, bye now.
Bye-bye.